Well, Tyler, this beautiful fall day here in Western Pennsylvania, I think it is fitting that today we finish off the fish.
Yes.
Never thought I'd have seen this day come.
It fits
And
because it's one
here
season
it
ends,
is.
another season opens, and the
Yeah.
season of fish is ending, and the season of tetrapods will begin.
Yes, sir. It's very exciting, but we have one more group we have to get
Yes.
before we do that. But before we do, welcome everybody to Life the Complete Story. I am Alex. I'm one of your hosts. And then that is Tyler, the one who does all the work. And I just show up and look pretty. Opinions vary. Most of the time. We'll see. But this, as I said, this is Life the Complete Story. We will start our episode as we always do. Probably be a little bit of a shorter episode today because, as we said, this is a very important group, but it's not a big group that we're going to talk about today. But we'll hit our nature in the news before we get there. Tyler, take us away.
All right.
This seems like an interesting one.
So today's nature in the news is about sustainable feeding. So one of the largest crops in the world is rice. Millions of people eat rice on a daily basis, if not billions.
Across the globe, yeah.
So a little thing about rice, it's very resource intensive to harvest. But we think we found the solution. We apply selenium, which is a metal. It's in that block in the middle of the periodic table.
It's a
transition metal.
Transition metal.
That's the word. So they apply basically a finite amount of selenium, very small amount, to.
Actually, it is a metalloid, I believe.
Mother trucker.
It's a non-metal. It is.
It's a non-metal.
Atomic number 34. It is between arsenic and bromine.
You know, I took organic chemistry in there. You
That's
think I knew?
Yeah, I don't know. But selenium. It's a very important micronutrient.
Yeah,
very important.
So what we're doing is applying selenium to these rice plants. I'm not sure if it's directly to the seed or if it's to like a sproutling.
Seems to be to the sprouts or like the young plants.
Gotcha. So they're using the selenium as kind of like a additive to the crop so that we're not using as much nitrogen fertilizers because they're kind of expensive to make.
Yes,
and they make a lot of carbon dioxide waste.
Yes, with all the, especially when you get runoff, it causes a lot of the problems that we've talked about as far as like red tides and dead zones in the ocean.
Yes,
it's been a while since we've talked about those.
But so the crops only use about 40 to 60% of that nitrogen fertilizer. The other is either washed away or just unused. The fertilizer has too much nitrogen for the plant to uptake. So with the selenium, adding it enables you to stimulates the plant's photosynthesis, which by 40%, which means it's able to uptake more of that nitrogen before it's run off into the soils.
Yeah, and with that, it makes the roots grow bigger and healthier. So then you get the organic compounds back into the soil that help the soil microbes do their thing, which then pulls more nitrogen in, usable nitrogen. And like I said, it decreases the amount of nitrous oxide and ammonia, which are, I believe, the unusable forms of nitrogen.
Yeah, because that would be like the NO2 and the NH4.
Yeah, those are your unusable forms of nitrogen. I believe the nitrates and nitrates are the ones that are the usable.
Yeah, nitrates and nitrites.
Yes, which are you remove that one hydrogen and it becomes an ionic form. Little chemistry that's not super important right now.
Yeah,
but it's up to between 30 and 50% increases its NUE nitrogen uptake exponent. Yeah. I'm sure it says efficiency, nitrogen use efficiency.
So
there we go.
Almost 1.5 times as effective.
Or else I saw it said reducing negative impacts from nitrogen fertilization by 41% and increase the economic benefit by 38% per ton of rice. And it was done using drones to just very lightly spray the rice with a suspension of nanoscale selenium. That's said cool. The direct contact doing it that way is far more efficient than it would be if it was applied directly to the soil. I'm guessing it's able to infiltrate directly through the plant tissues like
Yeah.
the green tissue, especially if it's a photosynthesis catalyst, or kind of like it speeds up
Yeah.
that photosynthesis. You would want it to happen where the photosynthesis is.
There'll be an exhibitor.
Yes.
But that's all I have for today's Nature in the News.
That's pretty cool. That could mean big things.
Yeah. Food.
Yeah, and a lot and more food. Say here you go. Rice cultivation accounts for up to 20% of global nitrogen use. So it can reduce their applications by 30%. That's going to save a lot of fertilizer, and
You
they
go from
grow
what, 20% of the global use to probably 15%.
Yeah, between
If I'm
15
doing
and
math
20.
right.
Yeah, it said between 15 and 20%. So it would decrease it a pretty good amount.
Yeah.
And it also makes the rice itself more nutritious because the levels of proteins, critical amino acids, and of course the selenium, which is important to our diet and animal diets as well, also increase. So it makes them not only more efficient to grow, but also healthier, which it's a double whammy of good.
Right. All right, Alex, that's all we have for this.
Yeah, that's
Let's move
pretty
on
cool.
into our next little bit. We're going to go on into our lesson here.
Yes, sir. I've already got pictures pulled up. I was ready.
There we go. Yeah. Alex hopped into our podcast call a little bit earlier than I did and was able to do a little prefacing.
There we go.
So today we're talking about Copteridgii, which is the lobe-finned fish, completely different than everything else we've talked about previously.
These are, as I said, the last group of fish. Then it has a completely different. Like if you remember, all the other fish we talked about were the ray-finned fish or the cartilaginous fish. So this is the third group.
Yeah. So if you would think of it this way, a ray-finned fish has like hands for fins, where like each of your fingers is a spine, or a ray, and then there's a webbing between them, and that's how they move.
Fin.
Yeah, that's their fin. On a lobe-finned fish, their fins are basically arms.
Yeah, they're much more muscular, more actual like tissue, like physical, like movable tissue, as far as like Tyler said, like an arm, where there's actually like muscles and skin. It's not just the rays that are moved by muscles that like the pectoral muscles, like the chest muscles.
Right. So, as I said, these are lobe-finned fish, and they have that. They're called limb buds, is where their fins kind of stem from. They arrived during the Silurian period, which is way, way back in time, about 425 million years ago. And these guys would be the ancestors to modern tetrapods, anything with legs, amphibians, reptiles, birds, and mammals, all come from these guys in one way or another. And they were the dominant predators of the freshwater ecosystems during the Carboniferous and Permian periods. So when they arrived during the Silurian period, they were marine predators, apex predators, but they were still predators. And then as you get more freshwater ecosystems, these guys were able to evolve and be able to use that freshwater as their habitat, which led them to become the apex predators in those habitats. And then these guys had a massive die-off during that Great Dying, which is the Permian extinction.
Permian extinction.
The Permian Triassic extinction. And they never really quite recovered. As you'll see, only about 2 classes of Sarcopterygii are still alive. That's Coelacanthomorpha and Rhipidistia. And Alex, why don't you tell us about the coelacanthiforms?
I will do so. Let me switch to my other tabs. Coelacanthomorpha, also known as the coelacanths. These are the oldest group of lobe-finned fish, originating during the early Devonian. I said that Silurian. This is just after the Silurian is the Devonian. So about between about 400 million years ago. Very long time ago. They were thought to have gone extinct during the Cretaceous along with the dinosaurs about 65 million years ago. But then a living species was described off of the coast of South Africa in 1938. So this has become known as the Lazarus taxon because this group seemingly went extinct in the fossil record. They didn't see anything, only for it to be rediscovered millions of years later. There are, however, only two living species of coelacanth, the West Indian Ocean coelacanth and the Indonesian coelacanth. Now, these are more deep sea fish, which attributes to why it took so long for us to find them again. They have very sensitive eyes, so they have a lot of rods that allow them to see in dim light. Those are the visual cells that see like light and dark. So mostly they see mostly in grayscale, which when you're that deep, that's not as big a deal. Just be able to see color. They do have very unique hearts. Their chambers are arranged in like a straight tube instead of the kind of typical, like, I guess like a plus sign or if they're two chambers like top and bottom for the chambers of the heart. So that is quite different. And their brain case is also very interesting as well because it is mostly fat. The actual brain is only about 1.5% of all the goop that is just chilling there in their head. And along with that, instead of a swim bladder, they have a single lobed, what is considered a vestigial lung that is filled with fat. And thus it serves a similar purpose to the swim bladder. But because that fatty lung is so large, the kidneys were actually relocated and fused into one organ instead of two. One of the most well-adapted species for its environment, so it has a very slowly evolving genome. So these coelacanths are quite literally living fossils. They really haven't changed at all in the last few million years. It's pretty much been, if you went back to the time of our ancestors and were able to find a coelacanth, it would look probably the same as it does now.
And that's kind of the same for a lot of these other things that are like super well adapted to it. Like if you think of crocodiles and
Yes.
sharks, two other living fossils that really haven't changed much from their original ancestors. I mean, sharks have, obviously, because there's some really freaky looking sharks. We covered them a while ago. Like the Helicoprion has the weird like ripsaw mouth. But anything else to add, Alex, to our little coelacanth discussion?
That's all I have for the coelacanth. If you want to take us into our next group, which is the lungfish.
Yeah, so I said we'd cover the group Rhipidistia. It's kind of a lie. Rhipidistia would be like anything that's not a coelacanth. So that includes like tetrapods too. But today we're only covering Dipnoi, which is the lungfish. That is the class Dipnoi. And they have three orders within, or three families within it, 3 genuses, and 6 species. We'll get that in a little bit. They're lungfish. They're able to breathe air. Some have as one species a single lung. Some have 2 lungs in their long, slender bodies. These are the closest living relatives to tetrapods. So a long, long time ago, not quite a lungfish, but something of the like, which we believe to be Tiktaalik, kind of crawled onto the land. Rather than having, teeth, they have tooth plates, kind of like a catfish, which has like that sandpaper plate. They use that to crush hard-shelled organisms, mollusks, and things like that. The South American lungfish, which is the purplish one in Alex's image, was able to develop feathery appendages during breathing, which I believe comes off of, where does it say it in my notes? Off their pelvic fins. And those are used to help in gas exchange for their eggs. And then through convergent evolution, lungfish evolved internal nostrils and a brain similar to modern amphibians. So convergent evolution, Alex, that's whenever one thing happens with one group, and then the same thing happens in another group.
At the same time, but they're not related.
But not together.
Correct, yeah. So it's not like lungfish and amphibians came from a common ancestor and they had the same thing. It was later on that they kind of developed the same traits.
Yes. Their lungs are connected to the larynx and pharynx without a trachea. So trachea is something that we have in our windpipe region. And I forget exactly what
That
it
is
does.
the windpipe. It
Yeah.
is kind of a cartilaginous collection of like stacked rings that connect your mouth to your lungs, kind of gives some separation with this. The pharynx is basically the back of your mouth, where like when you go to swallow, that is where the pharynx is at. So it separates the esophagus and the windpipe. And in this case, instead of having that longer trachea that gives them some separation between the mouth and the lungs are pretty much right there.
And these guys differ from other fish we talked about. So we talked about a couple of different fish species in the past who are able to kind of like gulp air and store it in like a modified swim bladder. These guys actually have like vascularized lungs with lots of air sacs and things like that, so that they're able to legitimately breathe air and not just like gulp it for later.
Even
And
to the point where Tyler's about to say is they still have gills, but they really don't work.
yeah, their gills are so far atrophied because of they rely on their lungs so much and they don't use their gills that in 5 of the 6 species, the gills don't work at all. And then, as I said, there's 3 families with a total of 6 species: the Queensland lungfish, the South American lungfish, the marbled lungfish, and then these last 3 all live in Africa. They're the gilded lungfish, West African lungfish, and spotted lungfish. So their families are kind of broken up off of geographic range.
Yeah, and the picture that I have is the West African lungfish.
Awesome.
Funky looking guy.
That's really all we have.
Yeah, and I will add one cool thing for the lungfish. They found in Africa in some areas, like freshwater ponds that will dry up seasonally. These fish are able to survive because they pretty much bury themselves in the mud and just slow down their
A form of hibernation, but not quite. I forget what the term is.
Yeah, but since they're able to breathe air, they just pretty much slow down their digestion and their metabolism is the word I was thinking of. And they just are able to survive solely because they can breathe like land animals do.
Some species actually exhibit like similar motion patterns to terrestrial vertebrates. So they'll like move like fins non-simultaneous, like you know how like in a fin fish, both pectoral fins move at the same time.
Yeah.
These guys will like move one then the other, like they're crawling on the seafloor.
Okay.
What else we got here? Obviously, the air-breathing lungs and the internal nostrils talked about. Yep, looks good. Looks good to me.
That's pretty neat. Saying about 420 million. So during that Devonian is when the last common ancestor is thought to have been between lungfish and modern tetrapods. So they've been separated for a long time, but this is in our tree, a tree of life. These are the closest ancestors to tetrapods. They can live quite a long time. There's one in the Shed Aquarium of Chicago that lived to be 109, and then one that currently, as of 2022, there's an Australian lungfish named Methuselah that was 90 years old, or estimated to be over 90 years old. I don't know if they know 100% how old it is, and it doesn't say where it's at, but they live quite a long time. It's pretty cool. Yes, that is the common.
This is Tiktaalik.
Yes, that is what is thought to be like the first land-based tetrapod.
Yeah. So they still have like fins, but they don't act as fins. They just haven't developed like proper terrestrial feet yet. So they still use like a fin to move around. But they have you can see the tail and the elongated body form that looks like a lungfish that we've just talked about. Obviously their mouth is a bit different, but this would be the common ancestor between, marine and freshwater fish to terrestrial vertebrates.
And yeah, that's all I got. So I was trying to read some more, but it was not quite what I thought it had been. So
All right, Alex, I'll let you do your most wanted first because I've kind of
yes,
been
sir.
rambling a little bit.
Yes, sir. So our most wanted for today is the spiny water flea. Not real fancy. Nothing super
Also,
crazy.
not really a flea, right?
I don't believe so. So these are native to Eurasia. They are hold on, let me get a different thing. Here we go. So they are actually
Brachiopods.
yeah, so they are a crustacean. They are not a flea because fleas are insects, right? Or
No, they're arachnids.
they're arachnids. Well, they're bugs, colloquially bugs. So they are a tiny little crustacean, less than 15 millimeters or half an inch long, native to the fresh waters of northern Europe and Asia, but they are believed to have been introduced into the Great Lakes of North America in like the mid 1980s, first in Lake Huron, and then by the end of the 1980s into all of the Great Lakes, probably through ballast water from shipping. And the problem that they are such an issue is that they have caused a decline in the zooplankton population because of predation. They eat other zooplankton and they have pretty much decimated the population of the native zooplankton, which is causing problems for the fishery, like the small fish that would eat the zooplankton. And then, of course, that continues up the chain and it outcompetes with species like the perch, which eats the zooplankton, and other small crustaceans, which would have eaten the zooplankton as well. And it is starting to be seen in other lake systems, not just within the Great Lakes. Yeah, so they consume, yeah, one spiny water flea can consume 10 to 20 prey organisms in a day, and because they are so small, they I would say they fall under that quantity, not quality, as far as reproduction. So if you think one of them, that becomes thousands of them, and you multiply that 10 to 20 individual zooplankton in a day, and it just ramps up. Some small fish are not able to feed on it because it does have barbs. You can kind of see the barb on the back of it. But as they get bigger, the larger fish are able to eat them. So the native fish are eating them, but I think they are reproducing fast enough to survive. Even their eggs can actually survive being dried out or eaten by fish. So it's almost impossible to get rid of them. But that is all I
I hate when
have
you
for
have these
these
small
guys.
pests, Alex.
Yeah, because
They're
it's something
annoying.
like this that can survive. It is just what do you do to get rid of it?
Yes, it's also well, but when we introduced them, we probably had no idea they existed since cell theory is relatively new in terms of science. Being able to use a microscope and identify things, I well past the introduction of these things.
Yeah, I would
Anything
say
else to
so.
add?
That is all I have.
All right, we'll hop over into our amazing species for the day. We're talking Neoceratodus forsteri, also known as the Australian or Queensland lungfish. These guys live in Australia, obviously, but their
Very
exit region.
yeah,
What?
very small area in
Yeah,
Australia.
very small region. Just 2 river systems, I believe, is their official range. In the Burnett and Mary River systems, although it has been introduced in other regions of southeastern Queensland, as well as other regions of Australia, it seems, which is good. That's what we want. They are relatively stable right now in terms of like a population trend. There's nothing really hurting them too much because we have enough things in place. And they are listed as endangered by the IUCN because of their range only being about 330 square kilometers and relatively fragmented within that range. They have a couple threats to them, mostly dams and water management. So like dams stop the flow of water, the fish can't move throughout the river like they need to, as well as, pollution from just, urban waste and agricultural runoff and invasive species such as, that's a lot of words, unspecified. Awesome. Good job, IUCN. They are being illegally poached for human consumption or as pet food. And then there are 5 facilities in Queensland that are officially used for aquaculture. So you can have them as an aquarium pet, but only from one of these 5 official breeders. And then for a little bit more about like what the actual lungfish is and not just its endangered status and everything like that. They're kind of long, about 1.5 meters in length. So is that 5 feet almost?
4 to 5 feet, yeah.
4 to 5 feet. And can grow over 40 kilograms. So that's like 80 pounds of fish.
Yeah, it's a big fish.
Yeah, they are relatively well armored per se. They have large scales, as you can really make out in the picture pretty well.
Yeah.
They have small eyes and those paddle-like pectoral fins, and their dorsal fin kind of starts midway along the back and goes into its caudal fin, making one like uniform fin. And they are typically like a brown to green in color, and they feed on frogs, tadpoles, snail, shrimp, earthworms, and other small fish, and occasionally plant material. And they are night spawners, so they spawn exclusively at night during their spring and summer in Australia, so August to December, because hemispheres swap seasons.
Yes. Yeah.
But that's all I have for our amazing species for the day.
Cool.
We will move on into our ecology encyclopedia. Alex, what do we have today?
So today we're going to get a little bit deeper into some biology. We're going to talk about some DNA replication. So it's a very important part of biology because it's how things grow and live. So just to keep it pretty simple, of course, your DNA is held in a chromosome. For more advanced species, it is kind of coiled up into that kind of quote unquote X shape that we see. That is just to maximize storage space, especially in eukaryotic organisms. So pretty much anything that's not a bacteria or like bacteria, virus, archaea, everything that we have talked about up to this point.
Yep.
It coils it up because has to store it inside the nucleus. There's only so much space, but when those cells divide to make new cells, either like when something is growing as an embryo to increase in size or just to replace cells that die, that are damaged, whatever, just to maintain the cells that are present in the organism's body, that DNA must be replicated while it does that. And to do so, it starts with a protein enzyme called helicase, which helps unwind the DNA from this double helix that many of us are familiar with, and it separates it into its 2 sides. And then using another enzyme called DNA polymerase. So DNA is made up of their nucleotides, which are the bases, which are paired. So adenine and thymine, or cytosine and guanine, are the 2 sets that are paired.
Your A's and T's and your C's and G's.
Yes, sir. And what the DNA polymerase does is takes those kind of just nucleotides that are just like kind of loose that have been made, and then pairs those with their corresponding, the base that's on the original strand, to make 2 new strands. So that original strand's like the template strand, and then it makes the second strand, and then over time, eventually, it gets through the entire set of DNA, and you have 2 sets of DNA, to which then they can split into the 2 new cells. But this, like I said, this is important because this is what allows life to happen. And this is also where you get some problems as well. In DNA replication, things like cancer can be caused by faulty DNA replication, or
it's not always guaranteed to put the right pair where it belongs.
Yes, and then usually there is a protein that follows after DNA polymerase. It's kind of like the check a protein. I forget what it's called. It's
I do as well.
another enzyme that basically checks the work of the original polymerase. But even that, sometimes things get missed, and that's what leads to things such as like birth defects during embryonic development. If there's during the meiosis, which is similar to mitosis, that's what makes the gamete cells, so the sperm and the eggs. So DNA is usually paired. That separates it into single sets. So then when the sperm and egg meet, it becomes the full paired set. But besides the point, if there's a bad replication, it can cause problems in some cases to the point where it actually kills the cell, or in other cases where it causes issues such as cancer, where the cell then starts to, there's the integrated controls within the cell are kind of overpowered, and it causes all kinds of issues, either with bad protein synthesis, because the DNA is what is used to synthesize proteins within the cells, and that's what allows our bodies to do what they do. So DNA is the code that is used to make every living thing. And that's why I wanted to cover this a little bit, just to give some little biology background of why this is important, but without going too crazy into it to confuse everybody.
Yeah, we're not going to get into PCR or anything today.
No, PCR and your leading and lagging strand, that's a little more complicated. If you want to look it up on your own, go ahead, and if you have questions, feel free to ask. But to keep it pretty simple, this is what DNA replication looks like, and this is what allows us to survive.
Yes. That's all I have.
That's all I have as well. I wanted to keep it kind of short and sweet and simple, so that way we're not getting too crazy, too into the weeds for the viewers and the listeners.
All right. Well, Alex, if that's all you have and that's all I have, we'll go ahead and wrap it up for the day. As always, guys, make sure to follow us on Instagram and TikTok at Life Complete Story, Facebook at Life the Complete Story, wherever you listen to podcasts, whether that be Amazon Music, Spotify, iHeartRadio. Still no Apple. Cannot figure it out.
Not for lack of trying.
Yeah. YouTube. Make sure to subscribe, leave a comment, do all the things for the algorithms. And send us an email at lifecompletestory@gmail.com for questions, concerns, your findings, things like that. And Alex, what do we always say?
As we always say, keep on learning and learn something new. You'll never know what you find interesting. I've been Alex, and that has been Tyler. Thank you for joining us on another episode of Life the Complete Story, and we will see you all next week when we finally get our way into tetrapods, and I swear it starts getting more interesting from here.
Absolutely.
Thank you, everybody, and we'll see you next time.